be let in. So think of this as the lipid soluble. They have a key card to get in. They can easily just scan it and head on in. Whereas ions and water soluble, they have to be specifically let in. How regulation is divided between what goes in and out of the membrane is either passively or actively. Now, just take a second thinking about what these two words mean. What do you think it means with respect to how something moves across the membrane? What might each require? Well, we can compare these two types, passive and active, to the act of, or to a hill and a ball. In a passive movement, we can compare this to a ball rolling down a hill. And this is for two reasons. The first reason is that the person, they can just watch the ball roll down the hill. So if they're just watching, it doesn't take any energy to do that. So no energy required. The second point, how this is similar, is that this ball is rolling from high to low very easily. You might not exactly know what I'm talking about yet for this, but you will in a few slides. High to low is passive. Active, on the other hand, can be compared to someone pushing a ball up the hill. Again, for two reasons. The first, person is pushing the ball up the hill. Well, pushing, that takes energy, requires energy. And what's the energy a cell needs? ATP. So active is going to require ATP. The second, we can notice it's going from low to high. So passive transport, as we've seen, takes no energy. It uses a concentration gradient. So a concentration gradient, what a concentration gradient is when there's two or difference between two parts. So if this is the cell here, there's cell membrane, we can see that the concentration over here is different than it is on the inside of the cell. So the gradient, that's when there's different concentrations. The bigger the difference, the higher the concentration gradient. So it uses this concentration gradient by going from a high concentration to a low concentration until there's the same amount on both sides of the membrane. I'll just write that down here. You'll see a lot brackets used. This is for concentration. So it's going from high to low. Because it wants to balance it out in the end. So here, if we look, we would see a few of these molecules going through to make it even on both sides. With respect to passive transport, there are a few different types that are used with the cell membrane. The first is simple diffusion. What diffusion means is it's the movement of a substance from a low concentration, from high concentration to a low concentration until that equilibrium is reached. That's diffusion. And it's often said to be down or with the concentration gradient. What that just means is that it's going from a high to a low concentration. It's following that concentration gradient. By doing this, by moving to that lower concentration, it's not requiring any work. It can just easily pass through. Here we see an example of carbon dioxide in the cell. Very great compared to the outside. Because it's in a higher concentration in the cell, it's going to be passively, no energy, just a simple diffusion through the plasma membrane out to the extracellular fluid. Now, what kind of factors would you expect to affect the rate of diffusion? So the rate at which the molecules are moving from inside the cell to out, or out to in. Well, remember, if there's an increase of temperature, that membrane becomes more fluid. So that can affect the rate of diffusion because it can allow these molecules to go through the membrane much easier. There's more room for them to wiggle through. So one that might affect the rates is the temperature. Same goes if it's too cold, or if the temperature is cooler, the membrane is going to be more rigid and stiff. It's not going to allow those molecules to pass through quite as easy. Something else that might affect is how big or the size of those particles. The bigger they are, the slower they're going to be diffusing across the phospholipid bilayer. The last thing that might affect is how great or how steep that concentration gradient is. The higher the concentration gradient, so the more extreme it is, the faster the molecules are going to want to get out of that high concentration to the lower concentration. As they start to reach the equilibrium, the diffusion would be much slower. The second type of passive transport is facilitated diffusion. This is the same as diffusion, but this time it requires some kind of channel or transport route to act as a facilitator. So to help that molecule or particle into the cell. Can you guess why it would need this channel? It's probably because of its size. Maybe it's too big. Or it could be polar or charged. Remember, these types of molecules cannot easily go through this bilayer because of those hydrophobic hydrocarbons. How do these transport proteins contribute to a cell membrane's selective permeability? Remember, we said selective because it gets to choose which goes through it. So if we have these channels, well, the first time it was selective is because it would only allow non-polar small molecules in. But if it's going to allow the big and polar or charged molecules in, how is it selective now? Well, it's still selective because those transport proteins, they are specific as well. So they are only specific for a certain nutrient or other insoluble molecule. Here we have one for glucose. So as you can see, it would be only specifically for glucose, fits very specific to glucose, and it would allow it to come into the cell. So it's closed, only opens for that one type of molecule. This is an example of another channel. In this case, we are looking at the sodium-potassium channels. Just to take note here, sodium ion is Na plus and potassium is K plus. They're both positive. These channels are made up from proteins. They can be opened or closed depending on the need for the cell. But remember, this is still passive transport, so it doesn't require energy, and it still flows by diffusion. That means it's still from a high concentration to a low concentration. Now, usually in the cell, inside of the cell, it's higher with potassium, and on the outside, it's higher with sodium. That's how it usually is. And then these channels, they would open, allowing there to be a balance. So it's still going to go from high to low. So when this asks what happens to the sodium channels, what happens to sodium when the channels open? The sodium, because it's higher on the outside of the cell, when those channels open, where do you think it's going to go? Inside the cell, yeah. So sodium would run inside the cell. Not run, but go into the inside, diffuse to the inside. What happens to the potassium when the potassium channel is open? This time it's higher on the inside, lower on the outside. So the potassium would be leaving the cell. Just to note here, those channels, they, depending on what the extracellular space's environment is, if receptors have been notified, they may tell the channels they need to be open or closed. But when they become open, they work by diffusion, going from high to low. The third type of passive transportation or transport is called osmosis. And this week you'll learn more about osmosis in your lab and see some real experiments having to do with osmosis. But osmosis is a special case of diffusion. It's the net movement or diffusion of water only. Specifically in biological systems, osmosis has to do with water. Water often requires aquaporins. These are membrane proteins that will allow water to go through. Because remember, water is polar, so it's not going to, those hydrophobic ends are not going to like water. However, in some cases, because water is so small, it might squeak through the membrane. But most often, it requires aquaporins, those membrane proteins that allow water to go through them. Having to do with osmosis, you often hear of tonicity. This is what determines the direction of water flow. Specifically, there can be 3 different environments: isotonic, hypertonic, and hypotonic, which we'll go through in the next few slides. With osmosis, because it is passive, we are still seeing water go from a high concentration to low concentration. So we can see here there's lots of water inside the cell, very few solutes, and then outside there's more solutes. So water wants to try to balance that out by osmosis. Osmolarity is the total concentration of solutes. So those are the molecules that are dissolved, or the substances that are dissolved in a solution. Water and solutes each tend to diffuse passively. So again, this means they're going down concentration gradient from high to low, which we can see here, very high concentration. Oh, sorry, here, a very high concentration. So more water is moving in to balance that out. In osmosis, water diffuses from regions of low osmolarity. So when we think of low osmolarity, that is when the concentrations of water is very high. It's going to diffuse from low osmolarity to high osmolarity. That's when the concentrations of water is low. So a little bit confusing here, a little bit of opposites, but it's still going, if you think about it from a water standpoint, it's higher concentration of water to the lower concentration of water. It's trying to balance that out. Here we're looking at tonicity. So with the 3 different environments they could be in, they could have a hypertonic, looking at the word, breaking it down. Hyper meaning high or too much. So this is when the extracellular solutes, or the outside of the cell, they're at a higher concentration than the inside. So all the water wants to balance that out, so they leave the cell. So there's a higher concentration outside. The water wants to kind of help alleviate that to even out. So all the water leaves. And an isotonic, iso meaning the same. And this, it's at that kind of balance enough. The same amount is inside the cell as outside the cell. So they just freely diffuse at a normal rate. Lastly, we have hypotonic. Hypo means that there's a low concentration. With that, the water is going to want to rush in. So a lower concentration outside of the cell, the water is going to rush into the cell. Now, I like to memorize hypotonic with thinking hypo going to blow up, because if there's too much water, it's going to blow up the cell. So if the outside has a very low concentration, then it's going to push all the water inside the cell and blow up the cell. Here in this figure, it's showing turgor pressure with respect to plant cells. It depends on the solution's tonicity. So with enough water over here, it's fine, but here it has lost its turgor pressure. So it's kind of in this hyper condition. There's not enough water outside of the cell, so all the cell's water inside of it leaves and it wilts the plant. But if caught quick enough, this can be restored by watering the plant. Okay, let's look at this one more time. So we have isosomatic or isotonic. It's that type of solution. The extracellular solutes are equal to the intracellular solutes. So here we have that balance. This is a good picture of normal red blood cells, water moving in and out normally. We then have hyposomatic or hypotonic solutions. Remember, hypo going to blow. That means that the extracellular solutes are lower on the outside than the intracellular solutes. So if they're lower on the outside, it's going to want to go from low to high. So all the water travels into the cell, and as this one depicts, it blew up. There's too much water. The opposite happens for hyperosmotic or hypertonic solutions. The extracellular solutes are then higher, so higher on the outside than the intracellular, higher on the outside. They want to go from low to high, so water leaves the cell, shriveling the red blood cell.